Go ahead, Romina.
Hello, hello everyone. Thanks so much for joining us today. It's a great pleasure for me to have Maria Jalbrzikowski joining us today. She obtained her PhD from the University of California, where she trained in as a clinical psychologist with emphasis on behavioral neuroscience. She completed her postdoctoral work in neurobehavioral genetics and developmental cognitive neuroscience. Since 2017, she has been developing an independent research program as faculty in the Department of Psychiatry at the University of Pittsburgh School of Medicine. She uses a developmental framework to examine relationships between genomics, neural circuitry, and psychotic symptoms to understand mechanisms of and risk factors for psychosis. She incorporates methods from developmental psychopathology, cognitive neuroscience, statistical genetics, and computational modeling to construct normative templates of neurodevelopmental to identify how aberrant trajectories may contribute to psychosis onset, identify neural mechanism underlying psychosis risk, individual differences in behavior, and link genomic variation to brain maturation in typical and atypical samples to identify biological pathways contributing to psychosis. In this talk, we are honored to hear Maria, who will present neuroimaging findings from the first study from the Enigma Clinical High Risk Consortia, showing the later transition to psychosis is robustly associated with lower cortical thickness in high-risk individuals. She will also discuss ongoing work in her research program and her working group. And with this, I want to welcome Maria to this exciting talk today.
Thank you.
And so people, sorry, just some housekeeping notes. If people want to ask questions, you're welcome to put it in the chat so as not to interrupt, and I'll try to find a way when to interrupt for all of you. And if you can keep it to the end, we can also have that in the end.
Yes, I'm totally happy to take questions. I did mention that sometimes I get in the zone, so please just say something, Romina, while I'm presenting to kind of alert me that someone's asked a question.
Sure.
So I'm really looking forward to this opportunity to present my program of research. Oh, is it okay if I go ahead and start?
Oh, go ahead.
Okay, sorry. And in today's talk, I'm going to first provide you with a quick introduction to the basic concepts of psychosis that my research program is built on, to get us all on the same page and introduce you to my overarching goal, my research program. Then I'll present a recent study we just completed looking at structural MRI measures associated with psychosis onset in an international cohort of participants at clinical high risk for developing psychosis. And then I'll share some converging evidence looking at neurodevelopmental trajectories associated with psychosis in another cohort that is at genetic high risk for developing psychosis. And finally, I'll finish the talk with some ongoing work as well as future directions I plan to take my work. So the psychiatric syndrome that I focus most of my research on is psychosis, which is characterized by a disruption in one's perceptions and thoughts. And this, where someone it makes it difficult for someone to differentiate between what's real and what is not real. And we all have beliefs that are a little odd. So I want to clarify that I'm talking about individuals that see, hear, or believe things that are not real. And these experiences cause significant distress or impairment. And there are three key points about psychosis that form the foundation of my research. First, psychosis typically emerges during adolescence and young adulthood. This is a distinct period of brain maturation and specialization. We also know that psychotic symptoms occur on a spectrum. For example, it's fairly normal to walk down the street and mistakenly think someone was calling your name. But at the other end of the spectrum are someone experiencing and hearing and responding to auditory hallucinations and speaking back to that voice. And then finally, I want to introduce you to the two types of cohorts I'll be talking about that I use to assess psychosis risk. The first group I'll be focused on is a group at clinical high risk for developing psychosis, or CHR. And this group is most commonly characterized by individuals who have recently started experiencing these sub-threshold psychotic experiences, but they don't meet full criteria for a psychotic disorder. And for example, they may hear whispers that are disturbing, but they're still able to recognize that the experience is not real. and can do some reality testing. The second group I'll be talking about is individuals with a diagnosis of 22q microdeletion syndrome, or 22q for short. And these individuals are missing a chunk of chromosome 22, and this condition puts them at significantly increased risk for developing psychosis. And when I talk about 22q today, I'll be talking about youth that fall within this condition that we call psychosis spectrum, where individuals have a range of psychotic experiences from subthreshold yet distressing psychotic symptoms to meeting criteria for a psychotic disorder. And my program of research is grounded in the neurodevelopmental model of psychosis. And this model was originally developed as the neurodevelopmental model of schizophrenia in the 1980s, and it's really evolved over time. And broadly speaking, this model proposes that psychosis is due to a number of deviations from typical neurodevelopment, such as excessive synaptic pruning, or reduced interneuron activity, and or deficient myelination. However, when you look at the majority of existing neuroimaging studies of schizophrenia and psychosis, the focus is on these group differences in adulthood. And this is myself included, so it's not, you know, we all have done, this is an important first step. And I also began my research career studying visual and auditory processing deficits in adults with chronic schizophrenia. And while I really enjoyed learning about the neurobiological basis that underlies psychosis, I thought, well, we need to identify these individuals earlier in the course of illness prior to the diagnosis of psychotic disorder. And also, if you're interested in studying mechanisms, a lot of the interesting antecedents that give rise to psychosis likely occur during this earlier phase. So that really led me to pursue what is now the most developed aspect of my research program that focuses on understanding how does development contribute to the onset of psychotic symptoms. And I use a variety of methods to do this, but today the talk will be mainly focused on cortical thickness, a measure obtained from a T1 weighted anatomical image. This is a coronal view of a T1 weighted scan, and you measure the length of the gray matter from the white matter boundary to the peel surface to get a measure of cortical thickness. Now, what's key to my research program is that you need a strong foundation in typical development to understand atypical development. And so a question I often first ask is, how are the normative developmental trajectories changing over time? And this is often best characterized by a nonlinear process. And here you can see when this is actually a typical pattern of development observed in cortical thickness. So for most graphs that I'll be showing you today, age will be on the x-axis and some sort of neuroimaging measure will be on the y-axis. And I'll usually plot the line of best fit with the lighter shading reflecting the standard error of the line. And here I'm plotting this well-established normative developmental trajectory of cortical thickness that shows there's usually an increase here earlier on, and then there's a significant decrease from late childhood through adulthood, leveling off through later adulthood. So after you get this trajectory, you can then ask, when specifically are the changes taking place? And to do that, what you do is you obtain the derivatives of the age-associated slope and compare that value to a null distribution. You say, well, how far does it fall from the null distribution? And for most studies that I'll be presenting today, if the amount was greater than 2.5 standard deviations away from the mean of the null distribution, we considered that a statistically significant change. And that's really marked by a darker blue. So if you can say, okay, well, darker blue means there's significant change occurring.
I have a question, if I can
Sure.
interrupt. Why are the standard errors so smaller earlier? Is less people involved, or is really a true effect?
There's more people involved, actually. So,
right, yes, more people, yes, more.
Yeah, so most of our cohort that we had actually was within this age range. So we did a really good job measuring it here, not so well here actually, but it's consistent with the previous literature.
Thank you, great.
Mm-hmm. So I also have a question here too. Given the, I think this measure of change per year, I'm amazed that it's so stable. That's really impressive. Give along the lines of what Romina was asking with the standard errors. What about sex differences in the rates of change as well? Because those are well known to deviate by sex. Do you have any thoughts on that?
That's a really good question. Usually females mature a bit earlier than boys, but I don't have the plots to show that right now. But in any of our, usually females have intercept difference. They have a lower cortical thickness starting off, and then their maturation occurs a bit earlier. And people attribute that to like girls act older than more advanced than boys earlier on. I don't know. And that I think is just a very fascinating, important study of research specifically for psychosis because of the difference in age of onset in sexes. Anything that I show you today, there was not an interaction between sex and any of the variables of interest. So no interactions between sex and group, or no interactions between sex and age. There were only main effects of sex, meaning that like maybe it was shifted downward in females. Okay, so, right, and also this is very stable at the group level. So there's one of the reasons I don't plot when you have a ton of data, like this is a very large sample over, I don't know, it's about 2,000 people, and the variability is so wide. So it is important to know that the within subject change is actually very variable. And then once you get these normative developmental trajectories, you can ask, well, how do these trajectories differ in a high-risk youth like CHR or 22q? And you can directly compare these slopes and see when they're actually different from each other. You can also ask, do these periods of change where significant change is occurring, is that period different in the high-risk youth? And for example, in this plot, not only do the slopes differ, but when the change occurring differs. So you see this never levels off in this high-risk youth. And then I think these questions are important because this is a time when you can actually really impact trajectories, and also it delineates a time where there might be greater vulnerability or risk for psychiatric disorders. And there are lots of other reasons that I think this research is important. So, I believe that identifying alterations in neurodevelopmental trajectories will lead to earlier detection and intervention of psychiatric disorders. So, for example, say we know this normative pattern of brain structure and it decreases with increasing age. Well, maybe we can longitudinally identify a kid when they're going off track, say here, when the brain changes are just starting and possibly before symptom presentation, and intervene earlier than later on here, when the brain abnormalities are greater and symptoms are potentially more severe. It's also plausible that kids with differing neurodevelopmental trajectories will respond differently to treatment. So it's important to characterize this heterogeneity associated with different trajectories. And we can also use these studies of neuroimaging and development to identify mechanistic processes through which brain development influences psychotic symptoms. And that might help guide future treatments or provide us with biomarkers for measuring treatment response. So I'm now going to turn to the study that I'll be spending most of the time on. That's the Enigma CHR study, where we looked at these neuroanatomic predictors of psychosis onset. And as I mentioned at the start of the talk, CHR individuals are most often identified by the presence of these subthreshold symptoms that have either just started or gotten worse in the last year. Now, approximately 20% of CHR individuals go on to develop a psychotic disorder. And though some behavioral features, like a drop in social functioning or slower processing speed, have been identified as risk factors in CHR youth, evidence for the presence of a neuroanatomic alteration associated with CHR who go on to develop psychosis is inconsistent. And this is likely due to a combination of challenges. It's a very heterogeneous sample, so individual studies often lack power to detect real effects. Furthermore, because it's early in the course of illness, the brain alterations are likely more subtle. And then finally, most of the time, these neuroimaging studies of CHR participants do not look at the effects of development, and so they might be undermining or obscuring important effects. So how can we overcome these different challenges?
So, sorry, Maria, Cindy was asking at what age high-risk group start to present symptoms?
Oh, good question. So the majority of CHR participants fall within the age of 10 to mid-20s. And it varies by study, but usually I think of these kids that are in late childhood, early adolescence, all the way through mid-20s. Average age of onset for a full-blown psychotic disorder is between 20 to 25 usually. So the way that we went about solving the problems that I just talked about was increasing the sample. You can do this by increasing the sample size and looking at a wider age range. And so the study that I'm going to present to you today was really based on a collaborative effort by more than 100 different investigators, and the number of CHR individuals who developed psychosis was 600% larger than any other previous study. And I was really able to amass this large sample size due to all of my collaborators, but also because of Paul Thompson at USC, who created the Enhancing Neuroimaging Genetics Through Meta-Analysis, or Enigma consortium. And really one of the main goals of this entire consortium is to harmonize neuroimaging data internationally and identify neural markers of psychiatric illness. Now, under his direction, there's 50 different working groups that have been formed. And I really think they're providing us with a ground truth of what's disrupted in psychosis. And this is just a little plot of, here's all the different sites across the world that are doing this. And this is actually a bit outdated, probably. There's always more people joining. And I'm also just one small little cog. I'm one of those little, like this Enigma CHR is only one little small aspect of this consortia. So in October 2018, in conjunction with Dennis Hernas at Maastricht University, we started the CHR working group. And over the past 2.5 years, we brought together and harmonized neuroimaging and clinical data from more than 150 investigators at 31 different sites. And I'll be presenting on those results today. And Romina is one of those investigators.
Thanks. I feel awful. I'm interrupting all the time. Anyways,
Oh,
Lani has a question, and she wonders, and this is a great question, I haven't thought about this. Are there sex differences in age of onset of subthreshold symptoms in CHR?
Oh, subthreshold symptoms
Yes,
in CHR. That's a good question. It seems like a good, obvious question that I don't know the answer to. I have to look at, we'll have to check into this after the talk. I'm not sure if anyone's actually looked at that.
I don't think anyone did. I mean, so yeah. Anyways, great. Thank you for the question.
Thank you. So we had 4 different aims in this study. We wanted to know, is there a structural neuroanatomical profile associated with individuals at clinical high risk for developing psychosis? And that's irrespective of a later outcome, whether they develop psychosis or not, just the CHR status. Then we followed up and said, well, what about those who actually develop a psychotic disorder? Do they differ from high-risk youth who do not develop a psychotic disorder and healthy controls? We then asked, are there age-associated neuroanatomical deviations in CHR who later develop a psychotic disorder? And then we wanted to know, how do these effect sizes, these group differences that we find, compare to existing Enigma studies of psychosis? And again, thanks to the power of collaboration, we ended up with over 1,300 controls and a total of 1,500 CHR individuals with follow-up data. Now, it's important to clarify the study design here. At the baseline assessment is when the participants are first ascertained in this study. None of the CHR individuals met criteria for psychotic disorder diagnosis. The individual sites at this point conduct a clinical assessment that determines, yes, this participant qualifies as high risk. And then at that point, the participant completes an MRI scan. We then follow these participants over time, and the CHR participants are usually clinically assessed again. And that is when what we call conversion status, whether they developed a psychotic disorder or not, is assigned. And so when the participant had the MRI scan, we don't know, will you fall into the pink group, the CHR converters, or the purple group, the CHR non-converters? And I'll be using that shorthand the rest of the lecture. So it's only later that we assign group status. So it's really, you can really get a prospective idea of what's going on. And you can see we have about 1,200 non-converters and around 250 converters.
Sorry, I just have a question about that last slide. So the conversion rate here, is that elevated, you know, with respect to the regular literature? It seems, you know, that you have about 1 in 5 converting, roughly. Do I have that right? 1 in 6? The total PS of the total CHR sample.
Well, it's interesting. I didn't present the people without follow-up data, but when you incorporate, and that's about 300 other individuals, and when you do that, that's about it's about an 18% conversion rate.
So is that, would that be high?
No, between I would say around 20 to 40% develop a psychotic disorder.
Okay, thank you.
Or at least I mean, that's what it does vary, but I would say on average it's about 20%. So because we're combining data from all over the world, we need an MRI scan that's common to collect. So we focused on T1 weighted gray matter structural neuroimaging. And so for the cortex, we use the standard atlas parcellation available in FreeSurfer and measure cortical thickness and surface area. And I'll be showing you these sagittal plots of the medial and lateral views. I'll also be showing you subcortical volume findings in coronal view. And so this first question, back to that right here, irrespective of follow-up status, are there group differences? And importantly here, age was regressed out prior to the analysis. I know that development is very important, but we just focused on group differences first. And we ran general linear models, including age squared, sex, and estimated intracranial volume as covariates. And I'll be showing you plots for cortical thickness on the left, surface area in the middle, and subcortical volume on the right. And for each type of measure, we have the left and the right hemisphere. Okay. And the top row here just refers to the F statistic, which just says, okay, is there an overall effective group here? And so the darker the purple, the stronger the effective group. And just visually, you can see that all the dark purples in the cortical thickness area. And I think that's important to point out because you actually see really widespread differences in adults with established diagnosis of schizophrenia and chronic schizophrenia. So the fact that we are not seeing something here at the earlier stages is really quite interesting as well. And then any region that still had a statistically significant effective group after correction for multiple comparisons, we conducted these pairwise follow-up comparisons to understand the directionality. And I'll be showing you the Cohen's d effect sizes now, these colors that reflect the strength of the effect sizes of the group comparison. So red is the darker red is going to indicate that healthy controls had greater cortical thickness compared to CHR participants. And here, again, you're seeing that there's just this widespread reduction in cortical thickness in CHR participants. Now, some people who don't do CHR work ask me, it's a reasonable question to ask, well, why do we even care about this group? You know, I thought you're caring about psychosis. But I think it's worth noting that other CHR studies, as well as population studies of youth, show that experiencing psychotic or psychotic symptoms or psychotic-like experiences when you're younger puts you at significantly increased risk for developing a wide range of psychopathology as well as functional impairments. So this widespread cortical thickness might be tapping into a more general biomarker of risk for psychopathology. So we then followed up and we said, well, okay, what about those CHR participants who later developed a psychotic disorder when they received their conversion status at follow-up assessment? And again, we ran similar general linear models, similar to the first name, and asked, is there an overall effective group? But this time our group variable is the converters versus non-converters versus healthy control. And again, visually, you can see, wow, all the dark purple is in the cortical thickness section. That's where the strongest effect sizes are. And then again, we did the same thing with the pairwise comparisons in any region that was still statistically significant after correction for multiple comparisons. And so we first see that in converters versus non-converters, there are a wide number of regions, but that are significantly different. But what's interesting is that only a few are different in converters, healthy controls, and converters versus non-converters. And so for that, it's the paracentral lobular, superior temporal region, and the fusiform region. So these are the only regions that were different in converters compared to the other two groups. And here are the bar graphs of these three cortical thickness measures. So you can see all three groups together. The healthy controls are in teal, the non-converters are in purple, and the converters are in pink. And cortical thickness is on the y-axis. And you can see that on all these plots, the non-converters really fall intermediate to the healthy controls and converters. And you really have this reduction in the converters. And so this is pretty interesting because also two of these regions that you might consider functionally relevant for perceptual positive symptoms. The fusiform is involved in higher order visual processing, superior temporal cortex is involved in auditory processing. And both of these regions actually have been heavily implicated in schizophrenia. So we next ask my favorite question, which is, what about the role of development in these regions that we see group differences? And we assess the interaction between age and group status using general additive models for these three regions. And what this does is it allows you to model nonlinear effects. And it also does not make you assume the same functional form, such as a linear effect for each group separately. They can all each have their own form. For these analyses, we did restrict our age range to 12 to 25, the ages we had the sufficient coverage in all groups. And here you can see that for the three regions, healthy controls show this characteristic decrease in all three regions. And you really see a slowing of growth in the early 20s. Now, just a reminder, these plots underneath the lines, I'm showing the derivative or rate of change. So you can see that early on, the rate of change is stronger earlier on, particularly for these two regions. For non-converters in purple, for the fusiform, they show a slower, steady decrease in fusiform cortical thickness with increasing age, and it doesn't plateau. For the paracentral lobular and superior temporal region, the age-associated slope is very similar, aside from the intercept difference, the group difference. So the age-associated slope is similar, but not, you do see that reduction. Now, for the CHR converters in the fusiform region, the age-associated effect was completely absent. You can see that the line is quite flat and there's no colors in the derivative plot. For the paracentral lobule, we also see that they do not show this age-associated change as well. There's really this absence of accelerated growth during this time, and they don't plateau off. So they're kind of continuing, just slowly continuing along. And for the superior temporal region, the age-associated slopes are actually quite similar. So these findings suggest that age-associated trajectories might be specific to some regions. Trajectory differences might be specific to some regions like the fusiform or paracentral lobule. And it's also important to point out for two of these regions, between the ages of 12 to 17, the group differences are more evident. So this really highlights the importance of thinking about developmentally specific risk factors that might be present at one age, but not another, or stronger at one age than another. So finally, for this study, we wanted to know, how do our findings compare to other Enigma working group studies that have been conducted? Since we did harmonize processing of this neuroimaging data, and this same protocol is used across the different working groups, we can take advantage of this opportunity and compare our results to already published findings across different Enigma studies. So for every group, we have the same measure of cortical thickness and surface area in subcortical volume. And so on this graph, this is one of the few graphs that is not aged on the x-axis. Each dot I'll be showing you on here is one of the brain regions from the atlas, okay? And what you can say is the pattern of effect sizes of the group differences that were observed in already published studies of schizophrenia versus healthy controls, is it similar to the effect sizes that we found? And what we found is, yes, there is a positive correlation between the Enigma schizophrenia study effect sizes and our CHR converter effect sizes. And what you're seeing here is actually all the strongest effects, basically the regions that showed the strongest effect sizes in schizophrenia versus healthy controls also showed the strongest effect sizes in converters versus non-converters. And so,
Maria, sorry, there's a question from Cindy, and I guess this would apply for all the studies, all the results you're going to be presenting. So maybe you can comment whenever you find it appropriate, whether there is any effect of drug use or abuse in these results by participants.
Yeah, that's a really good question. And thankfully the reviewer, individual investigators brought this up within the working group prior to publication, but thankfully the reviewers did not. And the reason we did not assess this in the beginning, because when the advice we received when starting off with the working group to establish feasibility was get enough measures that you can do a study, but don't get too many measures, because basically you want to get investment on the part of the other collaborators and investigators, but they don't want to, this is not their study, so they don't want to invest a whole lot of time in harmonizing and getting their own data together. So at the start of this study, we decided not to get other axis 1 disorders or drug use measures, but that's certainly something that we're interested in pursuing in the future now that we've established feasibility of the study. So that is a possible confound driving it. Now, I do want to note that that's in terms of drug use as in illicit substance use. What was interesting about this entire sample is only about 10% of the participants were on antipsychotic medication, and maybe about 20% were on psychotropic medication. So it's it was very unlikely that medication was driving our results, but we did additional analyses in our 90-page supplemental text that shows that when we removed individuals with that were taking antipsychotic medication or removed individuals who were taking psychotropic medication, all the results I presented to you beforehand remain the same. Okay, so we said, okay, the regions that have the strongest effect sizes in adult schizophrenia also have the strongest effect sizes in our study. We then showed this same thing occurred in Enigma, the 22q microdeletion syndrome, individuals that had a psychotic disorder versus individuals who do not. And this is really nice because you're really seeing this convergence across 3 different groups and really providing some strong evidence, scientific evidence, that we're tapping into something real with the cortical thickness abnormalities. So in conclusion, because the CHR converter abnormalities were found only in cortical thickness measures, you can speculate about several possible neurobiological mechanisms underlying these abnormalities. So thinner cortex could reflect abnormal synaptic plasticity or pruning, which has been implicated in schizophrenia. But I think what's really interesting is that more recent evidence suggests that intracortical myelination and or the expression of these myelin-related genes may be mechanisms underlying cortical thinning. So it might be this myelin that's sort of pushing the cortical thickness thinner as a normative developmental process. The age-associated differences also suggest several possible mechanisms. So it could be that earlier age of onset reflects a deterioration of this neural circuitry. Or maybe there's accelerated developmental decrease in cortical thickness that occurred prior to the age that we measured them. Finally, the third idea could be that perhaps if we think of these as normative decreases in cortical thickness that they need to go through as a process of specialization, particularly this accelerated period that you see in the other two groups, but not in those who develop psychosis, perhaps this is necessary to go through this process to, obtain optimal functioning and adult levels of functioning, and people who develop psychosis don't do this. Now, importantly, this study is cross-sectional, so we actually can't directly test any of those hypotheses, but it does point to the importance of conducting longitudinal studies. And then finally, just the fact that I want to highlight the group differences are robust. So we did all kinds of tests to make sure we did jackknife resampling, removing sites. We did mega-analysis, we did meta-analysis, we did it with this kind of pre-processing, with this kind of pre-processing. So, and the results remain consistent. However, the results were quite small. So it only accounted between 1 to 4% of the variance each individual region in group status. So that's important to keep in mind, I think, because we don't want to oversell how important our findings are for, risk detection. And then finally, I think this is what was most exciting to me, is that Dennis and I created this resource for many investigators to use. And we actually have 5 additional ongoing projects occurring right now, and many more planned. So I'm now going to briefly just touch on a longitudinal study of 22q microdeletion syndrome that looks if these developmental trajectories are disrupted in those with 22q and psychosis. And 22q is caused by this hemizygous deletion on the long arm of chromosome 22. And it occurs in about 1 in every 5,000 live births, making it second most common neurogenetic syndrome. And here's an up-close schematic of the deleted region. This region consists of approximately 90 protein-coding genes, many of them implicated in neuronal processes. And while the entire phenotype of 22q is incredibly fascinating to me, the point that's most relevant for this talk is that 30 to 40% of adults with 22q meet criteria for a psychotic disorder diagnosis. And so this really is like a homogeneous genetic subtype that offers an opportunity to investigate mechanisms underlying psychosis. And I've spoken about this here and there, but previous Enigma studies have already found that these group differences in cortical thickness in 22q participants with psychosis compared to those without psychosis are very similar to the pattern found in adult schizophrenia versus healthy control. So really the group difference analyses that I presented on with the CHR, that's already been done, and we're pretty confident that they have a similar pattern of effects. It's not identical, but. And so for this particular study, we were able to focus on differences in development in 22q with psychosis spectrum symptoms. And then the second question we asked in this study was, well, are these findings specific to psychosis psychopathology in 22q? And this is a smaller sample, but it's one thing to point out is that it's rarer. This genetic subtype is rarer, and so it is a bit harder to collect. And I want to point out that I am using data from my graduate school mentor, Carrie Bearden. So this is all data that she collected in her laboratory and then kindly gave to me to analyze. And so basically, you can see there's 3 different groups, fairly well matched in age. The average age is in the teens, and you can see this waterfall plot here that is the age at which individual received a scan. Each dot is a subject, and if there's lines connecting them, that means that they received multiple scans, and they're linked in with a straight line. So participants had between 1 and 5 visits, and there's decent coverage for all the groups across the age range examined. And just like the previous study, we used this standard desiccant atlas after data was processed through FreeSurfer. And the results were fairly consistent across the cortex. It was a widespread effect. So the next set of plots that I'm going to show you are just for mean cortical thickness and total surface area, not for individual regions. And when we looked at these developmental trajectories in the 3 groups, again, we saw the typical decrease in cortical thickness in healthy controls. And individuals without a 22q psychosis spectrum diagnosis showed a very similar pattern here in purple. But the 22q psychosis spectrum group, you see this, again, flat age-associated decrease early on. So there's steeper here. And then this period of continued decrease after these groups level off. And so it's a flatter slope here, but a steeper slope here. Okay. And then in contrast, the developmental trajectories were very similar for surface area in all three groups. And then we then said, okay, well, what about it's important to note that psychosis is not the only psychopathology associated with 22q. So approximately 40 to 60% of these youth with 22q are diagnosed with an autism spectrum disorder. So Carrie's study had comprehensive clinical information on ASD diagnosis for these participants. So we were able to ask, well, what happens if you split the 22q group up into individuals with and without an autism spectrum diagnosis? And again, the healthy controls here are in teal, and 22q with ASD are in red, and 22q without an ASD diagnosis are in blue. You can see that the trajectories are relatively intact. And if anything, then the non-ASD group looks like it has more of an accelerated growth in late teens and early 20s. But the surface area findings really tell a different story. Unlike the other two groups, the other two groups show this consistent decrease in surface area, but those with ASD really show this increase early on from middle to late childhood, followed by a decrease later on. And that was actually quite consistent across the cortex. So I just wanted to touch on this for a minute, because what I think is cool about this study is that you're really starting to see convergence on age-associated effects. And it also provides suggestive developmental mechanisms responsible for psychosis onset. So maybe this first age-associated alteration marks individuals with earlier onset in a more severe course. Or did these individuals have an earlier insult earlier on that led to this flattened trajectory? And then maybe later on, there's this accelerated development with reference to what's normative, or sorry, maybe they're not yet. Maybe there was accelerated development with reference to the normative timetables here. And then the second age-associated aberration is indicative maybe of a later onset group driven by a differing neurobiological mechanism like synaptic pruning due to inflammation. And this continued decrease while everybody else is plateauing. And then finally, we also this 22q studies show that there's a suggestive pattern of specificity for cortical thickness abnormalities in 22q psychosis. I'd like to finish off the talk with a few directions that I'd like to take my research. They really all have to do with psychosis risk detection from a developmental perspective, or understanding neurobiological mechanisms of psychosis through a neurodevelopmental framework. I first just want to, briefly speak about some of the foundation, how we're building upon this Enigma CHR working group. Several other projects are actually taking place, and as I said, to me, this is one of the most exciting aspects of the working groups, is having this resource to mine data. And once sites join, they have the option of opting in or out for each specific study. So once a PI becomes part of Enigma, they still have control over what they're interested in being, what projects they're interested in being in. And I think that's really, I think a lot of investigators really like that flexibility, that they're still, they can pick and choose what they're going to be involved in. We have 5 ongoing proposals right now, and I'll just highlight 2 of them. First, Shalala Haas and Cheryl Corcoran at Mount Sinai are looking at the relationship between structural neuroimaging measures and dimensional measures of psychotic symptoms. And they're examining the degree to which this relationship is consistent in individuals with 22q microdeletion syndrome. So not just categorical diagnoses, but these positive, negative, disorganized symptoms. The other project I'd like to highlight is led by Shinsuke Koike at University of Tokyo. And he's examining how well a suite of structural MRI measures differentiate between those who do and do not develop a psychotic disorder. And so I will spoil a little bit of his results right now, but given the small effect sizes that we observed, so far, we really don't do very well in individualized risk detection. So once a site joins and contributes data, the individuals from the site have the opportunity to submit secondary proposals and undergo a formal review process, and then get to access the data. We're also expanding the data, and that's super exciting, the data that we're harmonizing. So as I said before, we only collected baseline structural MRI data. So we're next checking to see if there are different progressive structural MRI changes that take place in CHR individuals who develop psychosis. We're also collecting additional psychiatric disorder diagnoses and psychosocial functioning measures. And we plan to see how this additional information might help us parse out the heterogeneity that's inherent in the CHR status. Finally, we're also working on setting up some pipelines to harmonize diffusion-weighted and resting state fMRI. So it's really exciting. I'm noticing the time here, so I want to make sure that there's time for questions. So, all right, I'm going to skip this next thing that I talk about, but I do want to finish off the talk with this, pointing out that I think Montreal has a novel approach to preventing psychosis. And I apologize in advance if this joke doesn't work, but basically, can Carey Price make the save and block psychosis? And I say this because my husband is a huge hockey buff, and he's like, you need to put this slide in. So he specifically made a slide for me. I apologize for the cheesy joke, but I also wanted to say good luck in the playoffs tomorrow, because I hear it's a very exciting time for you. So in all seriousness, hopefully today I've shown you that using a developmental framework will help us improve our ability to understand mechanisms of and eventually risk factors contributing to psychosis. In particular, I showed this convergent evidence of flatter age-associated cortical thickness slope in late childhood and steeper age-related slope during late adolescence. This is associated with psychosis onset in 2 separate samples. And finally, despite these exciting findings, right now, we really aren't able to use neuroimaging measures from these individual brain regions as objective markers of risk. But I do think we're really at a unique time to think creatively about how can we make neuroimaging measures work? Can we obtain better signal from the noise through different, high dimensional modeling techniques? And I also think it's important in terms of clinical and behavioral assessment to think about other ways to assess psychosis risk. For example, work by Jai Shah and his colleagues here at McGill show that approximately, and others show that between 30 to 50% of individuals experiencing the first episode of psychosis do not experience these subthreshold symptoms associated with the CHR state. So I think the CHR literature has done a wonderful job, and we've really established this really nice framework in the field, but I think it's now time to expand and think creatively about where can we look next? And with that, I'd like to thank you for listening to the talk. There are many people to thank, but I really just want to highlight these Enigma CHR working group members. The data that I presented would not be possible with everybody's contributions. And in particular, the co-chair of the working group, Dennis Hernas, has been fabulous to work with. And then finally, I'd just like to point out my 3 wonderful mentors, Bernie Devlin, Bea Luna, and Carrie Bearden, who have helped me grow into the researcher I am today. There's a whole lot of other individuals, sources of funding, and I really appreciate the participants and their families. So thank you.
Thank you very much for the lovely talk and for allowing people and students to ask questions. I will pass on the baton to Malar. I have to leave in 7 minutes, and I know he has a question as well.
Okay.
And many others may have other questions. So I'm looking forward to the discussion as much as I can be part of. And thank you very much, Maria, for this wonderful talk.
No problem.
Hi, Maria. That was a great talk. Really, elegant work and really well articulated.
Oh, thank you.
I had a quick question just about how you estimated the slopes, I guess, in the Enigma sample. Can you walk me through that a bit? And if you've mentioned it already, I apologize.
Oh no, I don't. You know, I should have also said I egregiously jumped over the methods. So basically, maybe I should end the show first, and then. So we use this approach called the general additive models. And basically, I can show you the plots of the Enigma study, but I don't have a good slide to.
So you're using GAMS and then
Yeah.
in a multi-site framework, right?
Yes.
So did all this data go through something like Combat first, or like how did you because we've done similar things, but it gets challenging when you're in a multi-site kind of framework, right?
Absolutely. So good question. So we did use Combat prior to analyzing all our data through the GAMS. We include site as a covariate. We did do follow-up, just look if there were any interactions between site. And then we also ran other analyses where we didn't correct using Combat, and actually they held up, like the results hold up pretty consistently. And then the other thing we did was for all the analyses, both the group differences and the age associated ones, we did the jackknife resampling to see are any sites really driving this effect? And we found that in these regions, when less than 10% of the sites were removed, did the results change. So we had reason to believe it was pretty stable. That said, I think that, there could be, I think when we get to we can estimate these more group averages, but when we get to the individual level, the side effects really, really hinder us at that point. Detection.
Thanks.
I don't know if that answers your question, but.
Pardon me? No, it does. No, I'm looking forward to maybe having a deeper dive into it. It's always, and it's come such a long way how to deal with these multi-site samples. It's, it used to be that, the reflexive, you can't combine data from different sites. And I love that we're now at a place where, that's basically a solved problem, which is really nice.
I don't know if it's a solved problem. It's a dealable problem maybe, and one that like.
It's never going to be solved, but in the sense that we have good methods for approximating how to deal with that. I guess maybe that's a better way of articulating.
Yeah.
That. Fair enough. And the aforementioned Jai Shah has a question for you.
Hi, Maria. Thanks for a great talk. As I've said to you before, I'm particularly impressed by the ways in which you sort of pull together neurobiological data with an appreciation of things like phenomenology. So that's always a high point for me when I go to talks, and it can happen. So I appreciate that. I had a question for you, which maybe goes hand in hand with the earlier question that it might have been you, Millar, who had asked about conversion rates. And Maria, I suspect from the data that conversion rates themselves have changed over time. So with the initial cohorts, conversion rates were approximately 40%. Now it's closer to 20% with successive cohorts as they're being reported. So I'm curious about whether you had examined that with respect to the imaging data you have, sort of the whether it's which cohorts they were associated with, or whether there has been I don't know what one would find. It's a bit more of a shot in the dark, but might there be a parallel effect here seen at a neurobiological level?
Oh gosh, that's a good question. And we actually did not examine that. And I think, the way I'm thinking, we do have site effects of conversion rates. So the individual sites on their own really range from like 0% to, I think, 50% in this one group. And I was like, are you sure about that? Like I even followed up and asked them when they showed me the data. And so we could maybe plot just conversion rates by the time at which the data were collected, to first start off by that, and then see if we could somehow look at that from a neuroimaging perspective. Because some people, the data that they gave us is really, really old. Like, I mean, it's much older. It's like from 1999, 2000, when CHR was first coming out. Whereas, you know, data from like my study came from like a year ago. So that's a really good idea.
The reason I ask is, right, there's different explanations for this drop in conversion rate with successive cohorts. On the one hand, there, you know, are suggestions that maybe these are just different folks who were assessing at different periods of time. On the other hand, there was a question about, well, maybe it's a function of differential service provision. We, in theory at least, we know better how to provide services than we did when at-risk mental state services were initially pioneered. And one way to maybe get at some of those questions is to think a little bit about whether the same phenomenon is reflected in the neurobiology. So that's just where I was sort of coming from with that.
That's a really good idea. I think you could also, I think we are, you know, we are doing a very, what did I call it? I don't know, crude assessment of service usage in this additional harmonization of follow-up data. So we could also incorporate that to see, we didn't even assess in this first, like, if more people were receiving services, did less of them convert? Like, those are all questions that have actually gone unasked in the CHR Enigma study framework. So I think it highlights the limitations to what I just presented, but it also highlights the potential for opportunity that you could really take this data a lot of places. In fact, you just brought up something I hadn't even thought of, which is cool, because you stare at this data for days and you think you've thought of everything.
All good. So I have some other thoughts anyway, but happy to chat offline. It's of interest.
So we have time for maybe one more question. Are there any? All right. I guess given that there are none, it's a good time to maybe, thank Maria for such a wonderful talk. Really, really impressive work. I really like Jay said, I really love the convergence kind of between the biology and what's going on clinically here too, and trying to figure out kind of the parallels between schizophrenia and the genetic kind of variants that you were talking about too. So thanks so much. I really enjoyed that talk.